Hey folks, all right, we're going to go ahead and get right into it. So this is going to be about gene expression and regulating, okay? So at this point, you should be an expert on the idea of us using our DNA in order to go and make proteins. Basically, now we're going to go talk about how do we regulate it, right? How is that you turn genes on or you turn genes off? So that's going to be our focus on this one for today. All right, so there's all your different essential knowledges, basically talking about how you can have certain genes be turned off or certain areas of the chromosomes being turned off. And then we'll go into a little bit more specifics on how that ends up happening. So let's go ahead and take a look at this, right? So it says, what is a gene, right? It's a sequence of DNA that's going to encode for a protein. And genes are thought to be like light switches. They can either be turned on, meaning that we are coding for the proteins that are associated with them, and then, we can turn them off, which means they don't. So a question to think about is, are the genes on all the time? And if yes, can you turn them off? And if they're not all the time, how is it that you turn them off? And how they end up being accomplished. And this is basically how the things are regulated. You're going to go find that certain genes are basically on all the time, and then we turn them off when we don't need them, or they're going to be off all the time, and then they're turned on when you do need them. Wait, isn't that the exact same thing? Not really. It's kind of like there's methods in that, let's say that, if I'm saying like I need sandwiches, right? I'm constantly making me sandwiches. Constantly make sandwiches. All right, so you constantly are making sandwiches, and you're just constantly making sandwiches over and over and over and over and over and over and over again, right? No, not ever being turned off. But then we can turn it off, saying like, "Hey, we are at like 100 sandwiches. Like, no, stop doing that. You're always making a sandwich without really being told to do so. But now I tell you to stop making the sandwiches." Versus we go over to the next location where they are going to be making pizzas, but no pizzas are being made, right? And then they end up getting a message like, hey, we need to go make pizzas. So then that's when they start to make the pizzas, right? So in here, we're constantly doing the thing until we're being turned off. Here, we're not doing the thing until we're told to do it, right? And that's really more or less like how your genes end up working. So it's kind of the thing about, well, what genes need to be on all the time and then turned off, or what genes need to be turned off all the time until they need to be turned on. So like for examples of that, I mean, you can like think about like enzymes, right? Do I need a digestive enzyme if I'm not having lactose? No. So that, the lactase enzyme is going to be turned off. That gene will be turned off until it's ready to be turned on. Versus maybe other ones where it's like I need to constantly be pumping out RNA polymerase. I want to keep on creating RNA polymerase until I have enough of it. So then that way I'm able to, you know, have enough to make all my genes or make all my proteins. So this is just basically what happens, right? We can turn them on or off at specific locations. So it can happen during transcription, which is typically like the most common one that we're going to go take a look at. On before or during translation, so when we're actually making the protein, or after translation, after the protein is being made. So what we're going to have is what's called a regulatory sequence. You're going to want to go ahead and jot this down, the regulatory sequence. Basically, there are sections of DNA that will say where the regulatory protein will bind to. If you think about a regulatory protein, what do you think that means? It's basically like, here's your DNA sequence, right? Here's your DNA, and then your regulatory sequence is like right here. So I'm not going to go and transcribe this gene if like there's like a protein in the way right here, right? So it's kind of like little blockers. So these regulatory sequences are actually sections of where this regulatory protein will show up. And most of the time is really cool is like these regulatory proteins actually show up within the DNA sequence itself. So it's like, okay, we're going to go transcribe and translate this gene for the first time. And then once you have all your instructions, now you're like going to be plugging the protein here to regulate the process. You're going to go and create the enzymes that you're trying to create with this DNA sequence. So there's lots of really cool little nooks and crannies happening in that. All right, so this right here is actually showing you what's called an operon. We are going to be very familiar with the concept of an operon. An operon is specifically only found in prokaryotes. Um, you do not use operons. This is only in prokaryotes. And what they end up doing is that there are these different regions. So you have a silencer region, where it's a silencer region is basically where you bind to in order to silence or prevent the message from being sent. Or it could be an enhancer, where we're going to go ahead and turn this on to promote the idea of it happening. So basically, these enhancers and silencer regions are just like sequences that will slow down or they will speed up the rate of transcription. And then from there, you have what's called the operator section. The operator is where the regulatory protein binds to. So the regulatory protein binds to the operator in order to prevent transcription of the gene. So you throw the protein on there. Basically, RNA polymerase will go over here, and it's not going to be able to bind because it's going to be blocked at this point. Really, it's going to go and join onto this promoter region. This is the first beginning region that any DNA sequence happens, and then boom, off to the races, and then we end up getting the whole gene, right? And then boom, like take a look, the blue and the red is the only stuff that's going through there because that's actually the gene. All the yellow stuff is all the regulatory sequences that we are going to be using as well. All right, so you have this idea of epigenetics. This is actually something you need to make a note of. I've seen this like 2 out of 3 years. A question about this has always come up, so we're going to go make sure we give it the attention that it needs. Epigenetics is just basically reversible changes to the DNA or the histones. A way to look at it is that your DNA is set up like this, right? So here's your chromosome. Your chromosome is going to be in these supercoils, and then you put them in these coils, and then we wrap them around these histone proteins, and basically this is how you store it, right? So what we'll do is that we'll just kind of condense the DNA into these supercoils when we need it, and then we will reverse it or we'll relax it so that way we can actually get inside here, because you're not going to be able to do any transcription when you're like wrapped around these histone proteins. That's not going to go and happen. So instead it has to be this other stuff. I'm in the middle of recording a lesson. What's up? You have Mr. Coughlin's autoclave, and we're going to need it for a biotech project. They want me to get your licensing for it. Yeah, I'm in the middle of a thing right now, so can we like talk in like at the end of the day? Yeah, sure. All right, are you a bus or a car rider? Bus rider. Coming here at 2 o'clock. Okay. All right, cool. Thanks. Sorry about that. Well, the show must go on. Here we go, folks. All right, so at that point, what's going to end up happening is that we have this DNA. The DNA, we're not going to be able to access it if it's still wrapped around these histones. So what we do is we unwind the histones, or we remove the histones. They're just basically proteins that, like, kind of think like toilet paper rolls. It's kind of what they do. You wrap your DNA around them, like you wrap them around so. And then you just wrap them up into these like coils and stuff, right? What we do is that we just kind of, unwind them so that way I can get access to the genes right there. But then epigenetics basically says that, well, you're able to, you can roll up the toilet paper and you can unroll the toilet paper, right? So it's basically saying you can do one or the other. So if you're like constantly like needing a gene, right? Okay, cool. We'll go ahead and like have this be available for us, right? But if not, we'll just consistently have it like packed away until we actually need it, and then we can unwind the DNA, get the sequence that we need, wrap it back up, and call it a day. Pretty cool process. All right. So that's going to get us to your phenotypes, right? So this goes back to genetics. So phenotype is the physical appearance of what the gene ends up looking like. So it's like you are big B, little b, but you are coding for brown eyes. Or you're big T, little t, you're coding for tall. If you're little r, you're going to be smooth instead of being round, or whatever it ends up being. So basically, the phenotype is actually us seeing that the gene is being transcribed and eventually translated. So when we're actually looking at our phenotypes, what this is showing you right here is we can end up having like our cells, right? So think about it like this, right? So you have a cell, right? This is like what you start off with, right? When sperm and egg fuse, right? When they fuse, you now create a zygote. Well, how is it that we specialize into, information in order to become a neuron or to come into an epithelial cell? Well, there's actually regions inside of the DNA that are going to be in charge of actually doing that, right? This image right here is just showing us how in a section of a chromosome, section of specifically the developing, fly larva, that we can end up finding certain sequences coding for certain regions of it. You can remove those sequences, and then you can see what ends up happening. So if I end up removing this DFD gene that's located right here, well, cool. Like your fly is not going to have a mouth in order to go ahead and eat anything. So they've done this to be able to see where all these different genes are, these phenotypes, that they're actual things. So what we're saying is like your DNA, right? Like if your DNA is coding for, the mid legs right here, right? That information here is going to go and code what this leg is going to go ahead and look like. Why? Because that's actually what the gene is coding for. It's how it's going to go ahead and be developed. Um, so if we end up putting, I don't know, some regulatory protein on there that prevents this from working, well then like your whole fly is going to be missing like this part of its body at that point. So that's kind of like the power of genes and genetics in a nutshell. Right. This is us mentioning about the operons, the ones that you are going to need to be familiar with, which we are going to be spending a lot of time on them, which you need to know about this, is the lac operon and the trip operon, an inducible system and a repressible system. Basically, what it means is it always on and we turn it off, or is it always off and we're turning it on? That's literally what we're going to go and take a look at right here. So let's go ahead and take a look right here. Okay, so here is showing you your lactose being present, what ends up happening. Okay, so here is our, here's going to be our inducer. Okay, so here's our inducer. This inducer is attached to this protein right here, and it's an inactive repressor. So what ends up happening is that this is going to bind to the repressor area. So this is going to be over to the operon right here. And if this is blocked in here, right, that means that we are not going to be able to transcribe this gene. So that means it's not going to ever be transcribed to go and create all these eventually these different proteins to break down lactose. And basically what happens is that if lactose is lactose ends up being present, what we're going to go do is actually pull the regulatory protein off, because it's basically like, hey, there's lactose here, so what we need to go do is remove this repressor protein so we can actually go and break it down, right? And as long as lactose is present, we're going to continuously transcribe this gene over and over again until I make enough of these proteins to break down those lactose molecules right there. So in the case here, what that does is that it basically the ingredient is basically turning the process on or turning the process off. So with here, we are inducing the process by it always being off and us turning it on. Subsequently, if we're looking at this one with the tripped process, so here we have the DNA sequenced in RNA, and we have this inactive repressor protein, right? Well, this is always going to be on until we end up getting the tryptophan being available, and then once that ends up being available, then it binds to the operator, and then it's preventing transcription from taking place then at this point. So your differences between these processes is that the stuff will turn the gene on, or the stuff will turn the gene off, is a way to kind of like really, really, really simplify it. So in induced systems, the stuff will turn it on, and repressing, the stuff will turn it off. Excuse me. All right, so that's just kind of like, and this is going to be in eukaryotes. This is showing you eukaryotic gene regulation, which we'll go in a little bit more detail about. The promoter region has what's called a TATA box, which is just thymine and adenines kind of like all like together. It's a good promoter region. Our DNA polymerase will go ahead and attach to it with the other regulatory proteins, and then eventually it's off to the races. There's ways to activate it, repress it, speeding it up, slowing it down, and this is how we regulate our genes. Instead of it being like an operator that's going to be turning it on or off, we're basically able to speed it up or slow it down. There's not really going to be like a protein binding to an operator site. We don't even have operator sites. All right, so Kind of goes back to the questions: are the genes always turned on? Are they turned off? How are you turning them on? How are you turning them off? That's when we get into gene expression right here, or cell specialization. So if we go and take a look right here, right? So we have these different regions that are going to be necessary to turn certain DNA sequences on and certain DNA sequences off. So we say that there's like a hypothesis right here, right? So expression of the neuron-specific transcription factors and fibroblasts will turn the latter, into neurons. So here are some fibroblasts. We're going to go ahead and have these factors, these genes, and then eventually we'll go be able to pass it on to the next generation of stuff, right? What we mean by this, right? So what we're looking at are these promoter regions. Are we actually promoting the process of these transcription pathways actually taking place or not? So is this promoter turned on? Then I'm probably going to go and transcribe this gene. Is this promoter turned on? Boom, I'm transcribing that gene. This one, right? Now I'm going to be on that gene. And what ends up happening is that each one of these mRNA sequences are going to go code for a different protein. And now depending on your environment or your organism, sorry, I should say your cell, that's going to determine whether or not the genes are going to be transcribed or not. This is just showing us for blocking transcription. So you have your repressor binding proteins. Again, this is not going to be an operator, but it works just like it, where it's going to go ahead and bind to it, preventing it from DNA polymerase from being able to actually attach and make this happen. All right. And what are we actually taking a look at? Um, Depending on what the cell does, certain genes are going to be turned on or certain genes are going to be turned off. So we end up taking a look here, right? So they have a liver cell to the left and a lens cell, which is located in your eye to the right. If you don't need certain sequences, like let's say like albumin, right? This is a gene that's going to be used in order to help like have the liver do its job, right? Well, what ends up happening is that the control elements for the albumin are going to be turned on for the liver cell, but they're going to be regulated and turned off over here. We can see the regulator right there. But if I go and take a look at the lens cell, albumin is turned off because the regulator is right here. But instead, we're going to go ahead and turn this on over to here. So we're going to go and actually allow us to express this gene, so that way we're not having like random genes being transcribed that don't need it, right? So your DNA has all the information for each one of these, like, these genes, but only certain cells are specialized to actually be able to go and need them. So here we end up just having that there's some RNA that can disrupt your gene expression. So after transcription, I can just basically throw like an mRNA sequence, this microRNA sequence that ends up attaching to the mRNA, preventing it from being able to be translated into a ribosome. So it's kind of like, there's a protein that's attached to it. So basically, we're preventing us needing, instead of us going to the genetic level of the DNA, we're just now going to go and affect the mRNA that's now floating around in our cytoplasm, and then we end up removing it there. You also have RNA ACE, I think this is what it's called, or restriction enzymes. That what the restriction enzymes do is that they break apart DNA or RNA sequences. They'll look for a specific nucleotide sequence, and they'll actually go and remove them, so that way they can actually like break apart the mRNA in case we really don't need a gene anymore. You actually want to go and destroy it, right? So what ends up happening is that we find that there are, we end up waiting for this experiment to take place. We find out the genes were end up being transcribed, and boom, I now have neurons. When before all I had were these earlier fibroblasts, which were just like kind of like precursors. So again, we turn the gene on because we realize like, hey, this is going to become a neuron, and then over a couple of days, right, because it recognizes that it is a neuron, we end up making it. And this happens for all your cells, whether it's a bone cell, liver cell, white blood cells, does not really make a difference. All your cells are basically have all the instructions for everything, and then you're only pulling whatever information is necessary. Imagine that you had like a huge textbook of every single classroom that you had, right? So you had like science, you end up having English and history and math. You have all that information, right? But you're only going to use your math in your math class. You're only going to be using science in your science class, et cetera, et cetera. And that's basically what this is saying, is that if this is going to go turn into a specific gene, it will end up turning into it, and it's going to use the specific genetics to make it happen. There's also the big thing about stem cells, where stem cells are like blank versions of cells, and what they do is that they can become specialized to become cells that are nearby it. So they kind of sense what is the cell that's nearby through chemical messaging, and from there that will end up changing up what the actual cell will turn into. So it's like, okay, cool. Like, you can end up regenerating skin or regenerating um, osseous tissue or what have you and stuff. So really, really interesting, cool stuff with it. Stem cells are basically like blank cells that can turn to whatever they need to. All right, that's that. I will catch you later.